molecule number 2 from dad, right? And if this was an actual human cell and I had the time and wanted to, or in the patients, right, I would draw how many more of those? 42 more, right? Because I have 4, so 42 more. Okay? So that's what we look like at the beginning of interphase and actually throughout G1. Okay? What's going to happen in S phase? What's going to happen in S phase, which I'm not drawing? I guess maybe I'll draw really fast, like here. Just in. What are we doing? We're going to be copying. That's this out. That's it. Tell me. No, I'm just kidding. That's not. Here. We're going to be copying. Let me make this same length as that. We're going to be copying. So through S phase, starting at the beginning of S phase, right? What are we doing? We are. This is S phase 1 phase. We're copying all of those DNA molecules. So at the end, here we're counted 4 in our own cells, 46. Here, how many, if we counted this, if you could actually count this, I made them really small. This is, you went from 4 to 8, right? In our human cells, we go from 46 to 92, right? And so by the end of G2 phase, we have DNA molecule number 1 from mom and another DNA molecule number 1 from mom. Yes? We have DNA molecule number 2 from mom and another DNA molecule number 2 from mom. What enzyme had to do that? What 3 enzymes well, there's more enzymes, right? But remember, we're skipping over some enzymes, right? Like topoisomerase, phrase. But what enzymes were responsible for making this happen? DNA, polymerase, DNA, helicase, and DNA, ligase. Yeah? All right, so that comes like this. DNA molecule number 1 from dad, number 1 from dad, number 2 from dad, number 2 from dad. What form is this in? It's in chromatin format. It's DNA double helix, right, in chromatin format, wrapped around histone proteins. We're still in chromatin format. Everybody good? Yes? So when I ask in my directions, right, both on the directions on the submission page, but also on your actual handouts, right, I do ask you to draw interphase. I ask you to draw interphase, both actually before mitosis as well as meiosis. Yeah. Okay. Good. Yeah. Okay. So let me make you a little. Now I'm not going to be writing out what's happening because oh my gosh, can you imagine if I wrote out everything? I'm just saying it. But you guys have that on my PowerPoints and also your handout, which I know you don't have in front of you, but I'm going to give it to you guys. Yes.
So interphase, just making clear that it's the same thing for both.
Yeah, interphase is going to be the same for both mitosis and meiosis. Yes. Yeah. Good question. Okay, so now we're not done yet. No. So you know what we started doing at the end of G1? What did we start doing at the end of G1? We started copying these centrioles. So and that happens throughout. By the end of G2, how many do we have of these? We have 2 pairs, 4 of them. We have these 4 centrioles. And again, what are these centrioles made up of? Are these microtubules attached to one another in a very, very specific fashion, right? In a very patterned fashion. And there's 27 of them. Yeah. Okay, so now let's talk about mitosis. So the first, what's it called? Phase or stage of mitosis is prophase, right? You can think of it as the new acronym is like passed my anatomy test, right? PMAT. But P thinks of it like preparation. You're preparing your DNA molecules to ultimately, eventually be separated and divided from one another. Okay? So I'm going to actually draw prophase in 2 separate images because there's quite a lot of things going on in prophase, right? So okay, one of the things that happens in prophase, early prophase, is that these DNA molecules that are in chromatin format, they're going to super condense. They're going to condense and become in their most compacted form, which we know as chromosome, right? So what happens is chromatin condenses to chromosome, chromatin condenses to chromosome, and as it's doing that, as it's condensing into chromosome format. The two identical DNA molecules, all the different identical molecules, like you have, these now 92, right? But kind of like 46 pairs, if you will, that are identical to one another. So chromatin condenses to chromosome, and then the two identical chromosomes, so I see this guy here and this guy here. What is this? That's these two here. They become attached to one another at something called their centromeres. See how I said don't confuse these C words, because centriole and centromere sound almost identical, right? But they're not, so please don't confuse your C words, right? So chromatin condenses into chromosome format, and then the two identical chromosomes attached to each other, forming now these two identical twin sisters holding hands. What do we call this now? Sister chromatids, right? So chromatin condenses into chromosome, sister chromatids are attached at their centromeres. That's like two, I guess this can be put into like one event or like two separate, like bullet points, whatever, right? And you do that also with these. So these two chromatins condense into chromosome format, and also the identical ones are attached at their centromeres. These are pretty equal in size, okay, and these are pretty equal in size. Because remember, the blue and the red, these blue versus red, these two are not identical, but they are very similar, right? That's why I'm trying to make them the same length. These two, this red and this blue guy, right, are not identical, but they're very similar. But however, this red and this red, are these identical to one another? Yes, identical. You just copied them. This blue guy, identical to one another? Yes, these sister chromatids are identical to one another right now. Yes? Okay, so that's two things that happen. Another thing that will happen is, at this point during mitosis, you're not making any proteins. All your concern is about, dividing your, ultimately your DNA molecules and splitting the cell. So you're not making any proteins, so you're not gonna need any ribosomes, right? And so you're not also gonna need the nucleolus. So this guy right here, the nucleolus, right? One of the, it's not a big point, but it's written in your PowerPoints, and so make sure you know that. That nucleolus disappears because we don't need it right now. Okay? Yes?
Nucleolus, same as the nucleoli, because in the PowerPoint says.
Yeah, because so because some cells actually have like a couple of nucleoli within their nucleus.
Oh.
Yeah. So nucleoli or nucleolus disappears. Yes. Did you have a question? Do you have a question?
Oh, yeah.
Yeah.
So if the blue ones are identical and then the red ones.
Well, not all blue ones are identical, right? But these two are identical, but not these blue ones. These blue ones and these blue ones are not identical to each other. Is that clear, right? Like, so this blue one and this red one, see how they're like very similar size, right? Because this is ultimately the DNA molecule number 1 and DNA molecule number 1. This one, like say this was your cell, like one of your skin cells, right? We have 46 DNA molecules, 46 chromosomes, right? Ultimately, right, you at one point were a one-celled organism called a zygote. That zygote, that one-celled organism, where did it come from? It didn't just appear, right, magically, right? It came from a sperm cell and an egg cell or a sperm cell fertilizing an egg cell. So the sperm cell from your biological dad, right, had 23 DNA molecules, right, 23 chromosomes, and the egg cell from your biological mom had 23 chromosomes, right? And the sperm cell fertilized the egg cell, and then whoop, there you are, there we go, like that zygote, that 46 DNA molecule cell, right? There, that's that new organism, right? And so 23 you have 23 pairs. Why are they pairs? Just like you have 23 pairs of shoes. If you have 23 pairs of shoes, like one Havaiana, one Adidas, one Nike. I don't know why I keep saying Adidas, but I'm not like particular to, right? One pair of like heels, right? They're similar, but they're not identical. Does that make sense, right? If you could magically make an identical copy of one of your Adidas shoes, right? And then another like identical copy of the other Adidas shoes, you have like 4, right? And like that, does that make sense? Yeah. So again, this blue one, this red one, and this blue one, these guys, right? They're similar to one another. These guys are similar to one another. Like the gene that codes for some kind of protein. Let's pretend mucin, right? The glycoprotein that we've been talking about that like is for like lupus, right? It's not on chromosome 1. can't remember now, 7 or, it doesn't matter what gene it is, what chromosome it is. But if the gene for mucin was on chromosome number 1 here, you'd have that gene for mucin, maybe a different variant though of it, also in this spot right here on that chromosome number 1, or DNA molecule number 1. Is that clear? Yeah? So similar, but not 100% identical because we have different variations of different genes. They're called alleles. Is that, yeah? Okay, cool. Yes?
Just a quick question.
Yeah.
So the central will also duplicate in the S space.
Yes, yeah, like so here, we've got 46, right? And then in S space, right, this, you end up with two copies of it. This, you end up with 2 copies of it. That, you end up with 2 copies of it. That's S space, that's that DNA replication that we're talking about here.
But the centrals are also duplicated?
Oh, the centrioles, this starts at the end, it's different. Like the centriole, making copies of itself, that's not, that's microtubules.
Okay.
Right, so it's a different, like when I say copy itself, it's not through DNA replication, right? It's making more of this protein, basically, this tubulin.
Oh.
Yeah,
Okay.
because microtubules are made up of
So it's just
tubulin.
making more proteins.
Yeah, well, more, yes, and then forming it into a centriole, right? And so that starts at the end of G1, continues through S, and ends at G2. Yeah. But like when I say copying in that sense, that's different than the copying there.
Yeah.
Something else is happening. Yeah. Okay. What do we have here? Okay, so chromatin condenses into chromosome. The sister chromatids are attached at their centromeres. The nucleolus has disappeared. All right, these centrioles right here, they are made up of microtubules, and those microtubules can start growing in length, right? Coming out of these out of these centrioles, you can have these. Basically, collectively they're known as mitotic spindle fibers, but they are microtubules growing out of these centrioles, right? And so there's going to have to be some microtubules growing out of here, 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 out of here. So imagine the purple and the green are basically microtubules growing out of these centrioles, okay? And as they grow out, as they are growing, imagine my hands right here, my fists are centrioles. And growing, and these centrioles are made up of microtubules, 27 microtubules here, 27 microtubules here. And then the 27 microtubules start like growing in length. They keep adding like more tubulin protein to it. And so imagine my hand going like this, my fingers, and imagine that my fingers kept growing. Like, I know it's a weird thing to imagine, but imagine my fingers growing. What would happen to my fist as my fingers kept growing? My hands would get pushed away from one another, right? So that's what's happening. So basically, what it says in my PowerPoints is out of the centrioles, right, the mitotic spindle forms, so grows, right? Because the green and the purple right there is the mitotic spindle or the mitotic spindle fibers, right? So centrioles start getting pushed away from one another, eventually getting to opposite poles of the cell. How? Due to that growth of the mitotic spindle out of the centrioles. Okay. This is still prophase, so this is still prophase. At this point, I'm gonna draw the centrioles on opposite poles of the cell, like here and here. Well, how did they end up? Because you have these mitotic spindle fibers growing out of the centrioles, pushing the 2 centriole pairs, right, apart from one another, okay? Okay, now what's going to happen, I'm gonna tell you what's going to happen. One of this set of centrioles, that's why I made them 2 different colors. One set of these mitotic spindle fibers, did I say one set of centrioles? I meant to say one set of the mitotic spindle fibers, these microtubules, is going to be attaching to your sister chromatids from each pole of the cell at their centromere. Okay, that's going to be happening. I haven't drawn that yet, but that's going to be happening. But for that to happen, you know what, actually this is gonna take me too long. I'm just gonna do the chromosomes now like this. Is that okay? Is everybody okay with this? This is this. I just don't feel like, bubbling in and like spending a lot of time drawing this. So let me just draw it like that. Okay, so what you will see, right, is eventually one set of these mitotic spindle fibers, you will learn in a moment, they're called kinetochore microtubules, right? is going to be attaching to the sister chromatids at their centromeres from both poles of the cell. For that to be able to happen, what has to happen first? What has to disappear, you guys? The nuclear envelope has to disappear, that nuclear membrane, right? Basically, the nucleus has to be broken apart, right? So one of the things that happens is the nuclear membrane, the nuclear envelope, right? The nucleus disappears. Any of those statements would suffice, okay? And now what can happen is these mitotic spindle fibers, which we're gonna call kinetochore microtubules, are going to attach to the centromeres of these sister chromatids from both poles of the cell. Both poles of the cell. Can you see what I did right here? Right? What are these purple guys? I guess I'll write this here. These are called kinetochore microtubules. Why microtubules? Because literally out of these orange things are made up of microtubules, and growing out of the orange things, out of the centrioles, are microtubules, right? These guys are called kinetochore microtubules. These are the ones that attach to the centromeres. What's a centromere? The centromere is just literally this thing in the center where the two sister chromatids are attached to one another. So the kinetochore microtubules attach to the centromeres of these sister chromatids from both poles of the cell. And then what's going to happen next is the centrioles and the kinetochore microtubules kind of pull in a tug of war from one end to the other end, from one end to the other end, right? But because, like, you know, can you imagine like a tug of war? Now imagine if in a tug of war, if like on one side there's one person, there's the other person on the other side, they pick up a rope, and in the center of the rope is a handkerchief, right? And this one guy over here like pulls the rope like a little bit harder in one split second, and then the other guy pulls it a little harder the other way, and kind of the handkerchief that's in the center of the rope moves back and forth. But if both guys are equally strong, right, what happens to the handkerchief? It ultimately ends up in the middle. Right? It ends up in the middle, right? So basically the job of the kinetochore microtubules is, in a tug of war, pull on the sister chromatids from one end to the other end, but eventually the sister chromatids are going to end up in the middle of the cell equator, right? And once that happens, you have now entered the next phase, which think M is for middle, it's called metaphase, right? So once that happens, we have entered metaphase. Is that clear? What about these green guys that I drew? What are those? Those are also microtubules growing out of the centrioles, right? However, they do not like attach to anything. Instead, they just keep on growing and sliding past one another. And when they keep on growing, imagine, you know, and these can be kind of in the center as well, but these guys keep growing and growing, and as they do, what happens is these are getting longer and longer. What's going to happen to the centrioles? Like, imagine my hands are already here like this, but these guys, and they're going to be called polar microtubules. Also, if you've learned this as non-kinetochore microtubules, I would also, what's it called? Accept that, right? But these polar microtubules, imagine my, centriole here, as they're growing and sliding past one another, what's going to happen to the centrioles? They get pulled, like they get pushed this way, which basically elongates your cell. It elongates your cell because this cell, by the end of mitosis, right, is going to be elongated. Who's responsible for that? The polar microtubules. Those are your polar microtubules. Why are we calling them polar microtubules? North pole, south pole, like we're elongating the poles of the cell. That's why polar, yeah? Guys, what are these kinetochore microtubules and polar microtubules together? What is this collectively? That is literally your mitotic spindle. That's the mitotic spindle that I know you've heard at least once before in an anatomy, right? Mitotic spindle fibers, right? Or at least hopefully you should have. So that's your mitotic spindle or mitotic spindle fibers. That's literally your these microtubules, kinetochore microtubules and polar. And you guys, in meiosis, we're going to have centrioles as well. We're going to have kinetochore microtubules, polar microtubules. And we just don't call it mitotic spindle, we call it meiotic spindle fibers. That's the same thing. It's just that it's not mitosis, it's meiosis, so we call it meiotic spindle. Is that clear? Okay. All right. So once these sister chromatids are pulled to the center of the cell in this tug of war due to these kinetochore microtubules, like pulling on them in a tug of war, then we have entered which phase? Metaphase. We have entered metaphase. And in metaphase, I mean, there's not really much to say about metaphase, right? We've got your sister chromatids are aligned. Is this kind of in the center? I tried to make it in the center. At the, let me make this is the same length. I gotta make the red a little bit of the same length as the blue one because these are very similar to one another. That's approximately equal, right? In size, approximately equal in size, yeah. Okay, so sister chromatids are aligned at the cell equator. And that actually can be visible under the like a light microscope, and that is referred to, when you can see this, as something called metaphase plate. Metaphase plate. Like you just add a few polar microtubules, just because it's not like they disappear or anything, so we just add a few of them. Okay, so we are at metaphase. Metaphase, think of middle, right? Sister chromatids are aligned in the middle. Questions? Does anybody remember what happens next from anatomy?
Anaphase.
Anaphase. And what does the A stand for? Right, apart and away. Right, the sister chromatids are first split apart from one another into individual chromosomes, and those individual chromosomes are then pulled away from one another. To where? To opposite poles of the cell. Okay, so in anaphase, I'll draw anaphase. You have split the sister chromatids apart from one another into individual chromosomes. You might be like, why are you drawing it like that? Because they're going to get pulled like that, right? And there's like the centromeres here, and they're going to be pulled like that, right? So sister chromatids have split apart from one another into individual chromosomes, and then the individual chromosomes are then pulled away from one another via those kinetochore microtubules. Kinetochore microtubules. These kinetochore microtubules are actually moving on to these, like with the help of these motor proteins called kinetochores. These complex proteins, they're actually moving this way, but for all purposes, you can just think about like the kinetochore microtubules are pulling these individual chromosomes apart from one another. So imagine, so the purple is the kinetochore microtubules, right? And imagine the movement, right? Because this is obviously a two-dimensional drawing, is this way, right? These guys, the kinetochore microtubules are basically now getting shorter. Right? Before they were getting longer, now they're getting shorter, right? Pulling the individual chromosomes to opposite poles of this cell. And of course, the centriole is helping with that. Okay? What else is happening? What do the polar microtubules continue to do? The polar microtubules, they continue to slide past one another. So these polar microtubules, they are not shortening. They actually continue to lengthen, right? And which continues to do what? Elongate the cell. That continues to elongate the cell. That's why I put the cell like elongated like that. Okay? Any questions? So A is for apart and away. Splitting the sister chromatids apart from one another into individual chromosomes, and away, pulling them away from one another. And then this right here, what is the final phase of mitosis is? Telophase. You can think of it as like kind of the tidy up phase, right? Because for the event that you have to know for telophase are kind of the reverse of what happened in prophase. Okay? So telophase, your individual chromosomes are now at the opposite poles of the cell, right? At the opposite poles of the cell, and you have one double set, I guess I should say, of chromosomes on this side, and an identical set of chromosomes on that side. Right? I'm not finishing drawing. Do these chromosomes remain in chromosome format? They no longer do. The reason you packed it into this condensed state is because during this tug of war thingy, If you didn't have it in chromosome format, if you had it in chromosome sister chromatin format instead, if you had it in chromatin format, they'd start like breaking apart from one another, right? Like noodles, right? So, but now we've already we've separated the two separate sets of 46, 46, or well in our case 4 and 4, right? To opposite poles, we've done that separation, right? Equally, hopefully, right? And so now the chromosome can do what? What did we do here? We did chromatin condensed into chromosome format. What can we do here? Chromosome will uncoil or uncondense into chromatin format, right? So chromosomes uncoil into chromatin, right? Chromosome uncoil into chromatin. Okay. What else? What do eukaryotic cells have around their DNA? What organelle do eukaryotic cells store their DNA in? The nucleus. So what has to reform? The nuclear envelope. So remember, they disappeared here in later part of prophase. Now the nuclear envelope or the nucleus reappears. This is still one cell, but eventually this will be 2 cells, and these cells are going to have to do their jobs, whatever job they have in the body. So they're going to have to make proteins. So they're going to need ribosomes. So what reappears also is the nucleolus reappears. Nucleolus reappears. And then you guys, you know how we have those centrioles, right? And then we have built this mitotic spindle. What was the mitotic spindle for? I mean, one, the green guys was to elongate the cell. The purple guys, the kinetochore microtubules, were there too, so they can attach to the attach sister chromatids to each other. Put the sister chromatids in the middle, and then to separate the sister chromatids into individual chromosomes, and then pull the individual chromosomes to opposite poles of the cell. So we're making equal, we're making sure that this one end of the cell gets 46. And 1 chromosome 1 from dad, 1 chromosome 1 from mom, 1 chromosome 2 from dad, 1 chromosome 2 from mom on that side, and the same copy on the other side. So that was the job of the mitotic spindle, right? And the kinetochore microtubules. Do we need them still? We don't need that, the mitotic spindle anymore, right? So do you still need the centrioles? Yes, you still need the centrioles, but what disappears, here let me just do a polar microtubule, what disappears is the mitotic spindle. So the mitotic spindle disappears, right? You no longer need the mitotic spindle. Right? Remember, you formed the mitotic spindle here in early prophase, now you disappear it, right? So the 4 events that are kind of like the cleanup of prophase, the reverse of prophase, are what? Chromosome uncoils into chromatin, nuclear envelope reforms, nucleolus reappears, and the mitotic spindle breaks down. So that's kind of that's the tidy up phase, the teal phase. Questions? Now, are we done? Like, do we have 2 cells right now? No, right? Yes.
So there are 2 nuclear envelopes that form?
Yes.
Okay.
Yes, exactly. Yeah. Do we have 2 cells? Did we go? Are we? We are done with mitosis. Actually, we are done with that. The portion of cell division that's actually mitosis, which is separation of the DNA molecules, the chromosomes, right, into 2 opposite sides. But we need to still split this cytoplasm, right? And so that's known as cytokinesis. And really, cytokinesis kind of starts towards the end of anaphase and continues through telophase. Right? So kind of towards the end of anaphase, you're going to end up forming something called this cleavage furrow, like this little invagination of the plasma membrane here. And that cleavage furrow will continue through telophase, right? Will continue to grow and grow, invaginate, right? Become deeper and deeper. And so by end of cytokinesis, imagine this going deeper and deeper and deeper, eventually this kind of comes together, and then you end up with 2 separate cells, right, that are identical to one another. When we're talking about identical, we're talking about, the DNA content. So they're identical to one another. Right, we have how many DNA molecules in chromatin format? We have, well, right in this cell that I drew is 4, but in your human cells, it would be 46. How many here? 4, which is the equivalent of 46, because I'm not drawing 46. And if you then now look here, actually, I need to make sure that I add a nucleator in this. Right, so by end of cytokinesis, we have these 2 daughter cells that came from this parental cell. Look at this cell and those 2 cells. Count their DNA molecule number. How many? 1, 2, 3, 4, which is the equivalent of 46 for us. 1, 2, 3, 4, 1, 2, 3, 4. Is this clear? This is why I also drew interface, and I want you to draw it, so you can prove to yourself how like this and these 2 are identical to one another. And why I'm gonna want you to do the same thing for meiosis, because meiosis, at the beginning of interphase, before meiosis happens, interphase, it'll look like that, but by the time you are done with meiosis 1 and 2, they're not, the cells will not look like that. You'll have 4 of them instead of 2. Any questions, you guys? Yes?
Did you say the mitotic spindle disappears or like dissolves?
Disappears or dissolves, either, yeah.